Hello everyone. In this video we're going to solve the International GCSE 921 skill subject biology paper 2B from the January 2021 series. So let's start. For the paper 2 there is a very big passage for question number 1, so I'm not going to read the entire passage. I'm just going to jump to the questions. 1A says which organism named in the passage is a producer. So let us first find out in which part they are actually indicating this question about. So the question is from this second paragraph, and if you see there's this things like plankton is eaten by crustaceans and then eaten by fish. So the correct answers would be option B. Option B was microscopic plankton. Planktons are trees and plants are always considered as the producer of any ecosystem. So that means option B is the correct answer for 1A1. So option B is the correct answer. Part A2 says which organism named in the passage is a tertiary consumer. So the entire thing flowchart was like plankton was eaten by crustaceans, and then they were eaten by the fishes, and the fishes were further eaten by humans. And the question wants you to find the tertiary consumer of the entire web. So this is the producer, this is the primary consumer, this is the secondary consumer, and humans are actually the tertiary consumer. So in that case option C is the correct answer. I hope 1A2 was clear. Moving to part B. Explain how blocked tubes leading from the mouth can lead to death in some animals. Refer to lines 8 to 10 in the passage. So I'm not going to read the lines from 8 to 10. I'm just going to use my basic knowledge that how blocking the tubes of mouth with plastics can actually lead to death. We know that from our mouth we actually input the foods. So if our entire mouth is blocked, it means that we cannot eat some things, so our digestions would be low. So obviously less food will be available in our body, and less respiration will happen, and less respiration would eventually lead to the less energy availability, and thus leading to death. And again, if it blocks the mouth area, indicating the fishes have very like they only can breathe using the mouth, right? So if that mouth area is covered with plastic bags, that means obviously they cannot breathe properly, and that means less gas exchange is happening over time, and less gas exchange means that the less oxygen enters the entire body and less carbon dioxide is removed from the entire body. So obviously the organism cannot respire aerobically, it has to respire more in anaerobic respiration, and thus there will be a buildup of lactic acid and less energy availability again leading to the death of that organism. I hope I have cleared all my points, so I'm just going to write out the answer. Blocked tubes leading from the mouth causes less food eaten, causes less food to eat by the animal, causes less food eaten by the animal. This leads to less digestion, which causes less glucose availability. Blocking of mouth also means that the animal cannot breathe properly, So less oxygen enters the lungs and less carbon dioxide is removed. All these changes results in more anaerobic respiration, which produces less energy and more lactic acid. So with this, our question number 1b ends. I hope 1b was clear enough for you all to understand. Moving to 1c. Suggest why bioaccumulation of microplastics is a problem for humans. So if we refer to actually the passage, we need to go to the passage for this part. This part is there. Microplastics are similar in size and mass of many types of plankton. These microplastics are not biodegradable and build up in the bodies of organisms that occupy higher trophic level in food chains. This is called bioaccumulation. So bioaccumulation is actually indicating that if fishes are eating microplastics thinking that they are planktons, because microplastics and planktons are same in size and mass, so fishes and crustaceans get confused with the microplastics and planktons. So if crustaceans are eating the microplastics, they will keep on adding in the food chains and they will keep on being at higher levels in the trophic chain, in the food web. That is, at the end of the food chain was humans, and indirectly humans would be eating those microplastics, and that is obviously a bad thing for our health. So these things are called bioaccumulation. So, and it indicates in our question 1c, it says that why bioaccumulation of microplastic is a problem for humans. So it's a problem because it increases the amount of plastic inside the bodies increases along the food chain, and humans are at the end of the food chain. That means they are taking the plastic at a higher amount and eating when they are eating fishes. Also, bioaccumulation would also cause less fishes to survive. That means more fishes would die and the fish population would decrease, leading to less fishes available for humans to eat. So that's why bioaccumulation are very problematic matter for humans. I hope my points are clear enough. I'm writing the answer now. The amount of plastic inside the bodies increases along the food chain. Inside the bodies increases along the food chain as humans are at the end of the food chain. So they are taking in plastics when eating fishes. Also, less fish would be available. For humans, as plastics would kill the fishes, thus decreasing their population. One C is done. Moving to question number 1D. Calculate the difference between the number of plastic particles ingested in a year by a fin whale and a whale shark. Lines 29 to 31st. Give your answer in standard form. So let us first move back to the passage and identify the numbers. 200 by fin whales in Mediterranean. So fin whales, whale sharks are actually ingesting 2000 and fins are actually ingesting 200. So we need to find the percentage difference. No, not percentage difference, just calculate the difference, only difference. So difference between number of plastic particles ingested. Number of particles ingested in a year, and that would be 2000 minus 200, and they were ingested in one day, and we know in a year there are 365 days, so we need to multiply it with 365 and you get a value of 657,000, and as the question stated that give you answer in standard form, it will be 6.57 into 10 to the power 5, and this is the answer for 1D. I hope the 1D was clear. I'm moving to 1E. Explain the effects of reduced reproduction on biodiversity. Refer to lines 31 and 32. So just using basic knowledge, we can understand that if a population start to reproduce less, that means obviously the number of gene pools will decrease, the population would decrease, and eventually leading that less causes fewer food sources for the entire other food chains, like because every organism is eaten by another organism directly or indirectly. So if one organism starts to reproduce less, it indicates that their population would decrease, and if that their population is decreasing, the organisms that would be eating on the reduced population would also decrease because there will be a disturbance in the food chain. So that's why it would obviously disturb the biodiversity along the food chain. So let's write the answer. Reduced reproduction decreases the population, which causes fewer food sources, thus leading to a disturbance in the food chain. So 1E is done. Moving to 1F. Suggest a benefit of using biodegradable plastic bags. We know that the term biodegradable means that our microorganisms can break them down and they are digestible by the decomposers. So obviously they won't start accumulating in the nature and will not harm any other animals or organisms by blocking their mouths and noses. So indirectly they are indicating that they won't harm the animals, other animals, and they are also sustainable for the nature because our decomposers can easily digest these particles. So let's write the answer now. They can be easily digested by microorganisms. by microorganisms. Biodegradable plastic produces less microplastics. Less microplastics, so less animals are harmed. So less animals are harmed. So with this, our question number 1 ends. I hope it was clear enough for you all to understand. I'm moving to question number 2. The urinary system is used in excretion. The diagram shows the urinary system with parts labeled A, B, C, and D. Which part of the urinary system contains nephrons? We know that kidney is the one that contains nephron, and label A indicates to the part of kidney, so option A is the correct answer. So 2A, for question number 2A, option A is the correct answer, that is kidneys contain nephron. I hope it was clear. Moving to part B. The urinary system produces urine. Name a substance found in urine. We know urine contains water, urea, and so many other ions and minerals. So as it has only stated that name one substance found in urine, we can simply write water. Moving to part C, a scientist uses this method to investigate the effect of drinking different liquids on urine production. Drink 600 cm³ of water in the morning. Measure the volume of urine produced during the next 2 hours. Drink 600 cm³ of 0.8% salt solution at the same time the following morning. Measure the volume of urine produced during the next 2 hours. The table shows the scientist's results. So it shows the volume of urine produced in the next 2 hours for both water and salt solution, and we need to find explain the scientist results, that why the actually volume of urine produced during the next 2 hours is less for the salt solution and more for the water. So it basically means that when the person or the subject is actually eating just water, the blood concentration decreases, because water decreases the blood concentration. So water decreases the blood concentration, and as blood concentration decreases, this change is detected by our osmotic center, that is the hypothalamus. Hypothalamus detects this change in drop in concentration of our blood, and hypothalamus then responds to this change by producing less ADH hormone. And less ADH hormones is actually a hormone that affects on the collecting duct of the urinary system. So less ADH means that our urinary system, the collecting duct of our urinary system, becomes less permeable to water. So collecting duct becomes less permeable to water. It means that less water will be absorbed from the urine, and therefore producing a urine with large volume. So let's write the answer down. More urine is produced more urine is produced when the subject is given water. Is given water, as water decreases the blood concentration, as is the blood concentration. This change is detected by the hypothalamus. Which responds to this change by producing less ADH less ADH from the pituitary glands. Less ADH makes the collecting duct less permeable to water. Thus, less water is reabsorbed producing large volume of urine. So with this, our question number 2C ends. C1. I hope the question number 2C1 was clear. Moving to 2C2. Give two other factors that affect the volume of urine production. So the volume of urine production can obviously be altered or affected by the temperature, because at high temperature we will produce less urine, at lower temperature we will produce more urine. And it is also affected by the food eaten. Like if you eat more juicy foods like watermelons or other fruits that have high water content in it, that would cause more urine production. If you eat more dry foods, then it will cause you to produce less urine. So food eaten is also affecting the urine production. And exercise, amount of exercise we do, if you do high amount of exercise, that means less urine will be produced, and if you do low exercise, then more urine would be produced. So exercise, food eaten, temperature, and other factors also might affect the urine production. So the scientists should have controlled the environmental temperature of the subject. So we can just write it down as temperature, and the food eaten by the subject, so you can also write it down as food eaten. So with this, our question number 2 ends. Moving to question number 3. Micropropagation is used to produce plant clones. The process involves growing explants in vitro. State what is meant by the term in vitro. In vitro means that something that is being produced or carried out outside the body. It could be in petri dishes, it could be in test tubes, or it could be in other incubators. So the answer here would be growing something inside a test tube. Inside a test tube. So with this, our question number 3A ends. Moving to question number 3B. The explants grow new roots and shoots. A student investigates the effect of pH on the growth of new shoots. The table shows the student's results. We can see that up to 6 pH, the mean number of shoots per explant increases, but after that the mean number of shoots per explant decreases. So let's move to the question first. Explain the relationship between pH and the mean number of shoots per explant. So we have already stated the differences, that up to 6 pH, the mean number of shoots per explant actually increases, but after that the mean number of shoots per explant decreases. So let's write it down. Mean number of shoots per explant increases as pH increases. Up to pH 6, above pH 6, number of shoots per explant decreases, indicating the optimum pH for the enzymes for Edexcel plants is pH 6. Indicating the optimum pH for the enzymes in of explants is pH 6. So with this, our question number 3B1 ends. Moving to B2, describe a procedure the student could use to obtain explants and produce these results. So we need to explain the entire procedure of carrying out an experiment. So there's nothing much to explain in the entire experimental methods. So I'll just jump on the writing part. The student can use a forceps to cut the explants. To cut the explants from the same plant to reduce genetic variations. And wash them in hypochlorite they should place 5 explants in different petri dishes, or maybe he should, actually there's this one student, so he should place 5 explants in different petri dishes containing agar, nutrients, buffer and amino acids. And let the explants grow for the next 1 week. Next 1 week, all the petri dishes should be kept in the same temperature with all the petri dishes should be kept in the same room with constant temperature, and we should make sure that all the petri dishes are containing or getting same amount of carbon dioxide concentration and same amount of light intensity to make all the variables constant, to make this a fair investigation. So let's write it down that all the petri dishes should be kept in the same room with constant temperature. Same amount of carbon dioxide concentration. And same light intensity. So after 1 week, they should count the number of shoots that are coming out of the each plants to count the mean number of shoots from per plant. So let's write it down that after 1 week, they should count the number of shoots and calculate mean. So with this, our question number B2 ends. Moving to question number C. Give two benefits of using micro-propagation to produce new plants rather than using seeds. We can say that by using micro-propagation, it's a very faster process, so we can actually produce a large amount of plants within a very short time. Also, during micro-propagation, the plants that are growing will be genetically identical to the parent plants, which means that we will get the exact same features of the parent plants in the new plants that are being produced. So the plants are all going to be genetically identical. So let's write it down. One benefit is that we can produce a large amount of plants within a very short time. And that second point would be that the plants are all genetically identical. So with this, our question number 3C ends. I hope it was clear. Moving to question number 4. The diagram shows a transverse section through a leaf. It shows the 4 layers under a leaf. That's layer A, B, C, and D. Part A says explain how layer A is adapted for its role. So we know that layer we can see that layer A is the uppermost layer of this leaf, and the uppermost layer, it always has to be transparent so that it can allow light to reach the lower layers, that is layer B, the palisade layer, and it has the waxy cuticle which reduces the water loss to give protection against like the waxy cuticle also provides the plant from excess water loss, and it also gives the plant protection against infections. So let's write it down. Layer A, as they didn't mention, they asked us to name the layer A, so we do not need to mention the name of layer A. So we're just going to write it at layer A is thin, transparent cell to allow light reach the palisade layer. The waxy cuticle it also has waxy cuticle. which reduces the water loss from a plant and gives it protection against infections. So with this, our question number 4a ends. Moving to 4b. Explain how layers B and C are adapted for photosynthesis and gas exchange. Let's first go to the diagram. We can see that layer B has a lot of chloroplast and they have a bigger surface area, so they can easily absorb quite a big amount of light, and as they have chloroplast, so they can also carry out photosynthesis. And layer C, we can see that layer C has very big air spaces between them, and these air spaces makes them more efficient for gas exchange, so carbon dioxide and oxygen can easily diffuse in and out of the cell. So these are the adaptations for layer A and B. Layer A is the palisade layer and layer C is the mesophyll layer. So let's write it down. Again, the question didn't ask us to mention the layer B and layer C, so we're just gonna address them as layer B and layer C. So first we will write the adaptations for layer B. Layer B has cells with many chloroplasts. And large surface area. To absorb the most sunlight. To carry out photosynthesis. Now moving to layer C, and layer C is the has cells with air spaces between them. Which makes it more efficient for gas exchange. So carbon dioxide and oxygen can easily diffuse in and out of the cell. So with this, our question number 4b ends. I hope it was clear enough. Moving to 4c, explain how layer D is able to regulate gas exchange. Let us first move to the diagram. We can see that layer D contains the stomata, and it is guarded by and it is covered by the guard cells that controls the opening and closing of stomata to let carbon dioxide in during the daytime and oxygen out. So obviously with the help of the guard cells, they control the opening and closing, which makes it adapted for the gas exchange. So let's write the answers. It has guard cell, that controls the opening and closing of stomata to let carbon dioxide in during the daytime. So with this, our question number 4C. Moving to question number 4D. Gardeners sometimes apply a spray called an anti-transparent to plant leaves. The spray is impermeable to water vapor but allows other other gases to pass through. Discuss whether an anti-transparent spray will promote plant growth. So as the question says discuss, it means that we need to present ideas both for and against the main part, that anti-transparent do actually promote the plant growth or not. So let's first write the points that are for the idea, and that is, if this spray makes the leaves impermeable to water, that means there will be a reduced transpiration. Reduced transpiration means that the leaves or the entire plant will lose less water now, and as the plant is losing less water, it can use more of these waters to carry out photosynthesis and produce food that will eventually promote the plant growth. So one point is that it causes reduced transpiration. Reduced transpiration saves up more water for the photosynthesis to carry out for the plant, and they can produce more food for their plant growth. And the other point for their plant growth is that this spray makes the leaves impermeable to just water vapor but permeable to all the other gases. That means carbon dioxide can easily diffuse in, and they can also use the sufficient amount of carbon dioxide for the photosynthesis to occur. That means again promoting the growth of plant growth. So yeah, these are the 2 points that are actually for the idea that anti-transparent sprays do promote plant growth. Now, as the question says that we need to discuss the ideas, that means we need to represent points against the entire motive too. So the against ideas are that if the leaves are impermeable to water, as we have already stated, that it makes the plant leaves it reduces the entire transpiration of the plant, and if there is a reduced transpiration, that means the pull of water will also be less. And we know that minerals and ions from the ground are received by the roots, and the minerals and ions are made to travel up towards the leaf by the pull of water. And if there is a reduced transpiration, that means the pull of water has reduced, and therefore the minerals cannot efficiently travel or cannot reach their destinations where they are needed. So even if the plant is given enough minerals, it still will not get enough. It still will not receive those minerals to their sufficient places. And again, if there is not enough minerals, then it might not cause or might not produce enough chlorophyll to carry out photosynthesis, and eventually decreasing the plant growth rate. So reduced transpiration causes less minerals to move up to the plants, and eventually it would reduce the rate of photosynthesis, and which would cause the plant growth to decrease. And the other point is that transpiration causes the plants to cool down. So if there is a reduced transpiration, it means that there is a chance of the plant getting excess heated up. So these are all the points that I have discussed. Let's write the answers now. It makes the leaves impermeable to water. So transpiration is reduced, leading to less water loss. So more water is available for the plant to use for photosynthesis to carry out to produce more food, or we can say that so the plant becomes less flaccid easily. So the plant does not go flaccid easily. as the spray allows other gases to pass through, so carbon dioxide can easily get in and photosynthesis can occur. So carbon dioxide can easily get in and photosynthesis can occur to produce more food that promotes plant growth. Now we will represent points that are against the idea. So the against ideas are reduced transpiration, would also decrease the transport of mineral ions and may cause the plants to overheat. All these will reduce the rate of photosynthesis so less food will be produced leading to less plant growth. And with this, our question number 4 ends. I hope question number 4D was clear. Moving to question number 5. 5A, DNA strands has this sequence of bases. How many amino acids does this sequence of bases code for? So we know that there will be 3 nucleotides for one amino acid. So in that sense, let me use a highlighter. So this will code for one amino acid, CAT. This will code for TCA, will code for another amino acid. ATT will code for another amino acid. CAT will code for another amino acid. And the least TTC will code for another amino acid. So we can see that there are 1, 2, 3, 4, 5 amino acid, indicating option C is the correct answer, that there are 5 amino acids for this basis of codes. So I hope 5A1 was clear. Moving to 5A2. Whether write down the complementary mRNA code for this sequence of bases. So there are CATTCAA, TTCATTTC. This was the base shown in the question, and we need to represent the complementary mRNA code. We know that in mRNA it has both the A nucleotide, G nucleotide, C nucleotide, but the T nucleotide is absent in mRNAs, and in return it has the U mRNA. In mRNA it has the U and nucleotide instead for the T nucleotide. So C stands for G, and for A it will be a U on mRNA. For T it will be an A, so there will be an A. For C again a G, and again U, A, C, C stands for G, and then A would be U, and then A, C. And with this, we have written the complementary mRNA code for this sequence of bases. So this was the answer, not this was the question. I wrote it just for my own use. This was the answer for question number 5A2. I hope it was clear. Moving to 5A3. A student reads the DNA codons are non-overlapping. So just what is meant by the term non-overlapping? You should refer to the sequence of bases in your answer. So let us move back to this page, and here we can see that it has CA, or maybe I can repeat it down on the next page. So let me move to the next page. The sequence that was given, it was CAT, TCA, ATT, CAT, and TTC. So the term non-overlapping means that if anyhow these threes will not overlap with each other to form a new amino acid. This will not gonna happen ever, that ATT will somehow combine with each other to form a new amino acid. This is not gonna happen because amino acids are very discrete. It has to be very specific 3 nucleotides to form one amino acid. It has to be very specific, very discrete, and that's why the different nucleotides, even though they are very close to each other and they are like one beside the another, even though there is very small gap between the nucleotides, still they will not combine together to form a new amino acid. Like ATT will never combine to form a new amino acid. Like AAT will never combine to form a new amino acid. Like TCA will never combine to form a new amino acid because the amino acids are very discrete. It has to be specific 3 nucleotides combined together to form a specific amino acid. So that's why overlapping is never possible for the DNA codons. I hope I have made my point. So I'm just going to write the answer. Each triplet codes for one amino acid. Codes for one amino acid, and all the codons are discrete. Codons are discrete. So with this, our 5A3 ends. Moving to question number B. Describe what happens in the transpiration stage of protein synthesis. So we need to write the entire thing in detail, that how mRNA arrives on the ribosome, and the ribosomes starts moving across the mRNA because the mRNA is acting as a template, and the ribosome then binds the anticodons on tRNA with the codons on the mRNA to form amino acids, and the amino acids are then joined together by peptide bonds to form the eventual result, that is the polypeptide chain. So let's write the entire process down in detail. The mRNA arrives at the ribosome and acts as a template. The mRNA arrives at the ribosome and acts as a template. The ribosome moves along the mRNA strand. And it binds tRNA with codons on mRNA to form amino acids. The amino acids are joined together by peptide bonds. To form polypeptide chain. And with this, the entire process for the description of transpiration stage is done. Moving to 5C. What is meant by the genome of an organism? We know that genome is basically the part which contains the genetic material of that organism. So just write it down because it contains only one mark. So genome means the genetic material of an organism. Of an organism. So with this, our entire question number 5 ends. I hope it was clear enough for you all to understand. Moving to question number 6. 6 says a catalyst is an enzyme found in many cells. It speeds up the breakdown of hydrogen peroxide into water and oxygen. The equation for the reaction is 2H2O2 gives 2 water molecules and 1 oxygen molecule. A teacher demonstrates the effect of increasing catalase concentration on the initial rate of reaction. This is the teacher's method. Cut 5 equal sized discs from a potato, each 0.2 millimeters thick. Place the discs in a boiling tube with 5 cm cube of buffer solution. Add 5 cm cube of hydrogen peroxide solution into the boiling tube. Place a bung and delivery tube firmly to the boiling tube. Position the other end of the delivery tube under our inverted measuring cylinder. Start a timer as soon as the first bubble of oxygen enters the measuring cylinder. And lastly, measure the volume of oxygen produced in 1 minute. So repeat the method 3 times to obviously calculate average. So this is the entire diagram showing how the entire apparatus is set up. The teacher uses this method with different numbers of potato discs, making sure the other conditions are unchanged. Part A says give the expected relationship between the named independent variable and the named dependent variable in this demonstration. So we know that the independent variable here, the main thing that was actually being measured was the volume of oxygen produced. So the increasing concentration and the amount of enzyme concentration there were. So enzyme concentration would be the independent variable, amount of oxygen produced would be the dependent variable, and the relationship would be obviously that increasing the enzyme concentration increases the rate of oxygen production. So let's write this down. Increasing the enzyme concentration increases the rate of reaction. Rate of oxygen produced. So we have stated clearly the dependent and independent variable and also the relationship between them, that like they are directly proportional. That's why when the independent variable increases, the dependent variable also increases. So part A1 is done. So we need to now give 2 variables that the teacher controls in this demonstration. The teacher should have controlled the pH, the buffer solution of the entire system, and also the volume of hydrogen peroxide that the teacher was using. So it says pH and volume of hydrogen peroxide. Moving to question number C, the table shows the teacher's result, and there is this one blank space. We need to find the blank amount, and that's the mean part. We need to find the mean reading for the first column. So let's write it down. Mean is equals to 1.2, 1.5 plus 0.0 plus 1.5 divided by 3, and it gives a value of 1.4. So it will be 1.4 in this blank. Let's move to the next part. Calculate the mean volume of oxygen produced in 1 minute using 5 potato discs, and this is exactly what we found in the last page. This is the answer for part 1, 1.4. And in part 2 it says calculate the percentage increase in mean volume of oxygen produced in 1 minute as the concentration of enzyme changes from 15 to 20 discs. So let us first find the amount of oxygen produced for both the discs. That is 7.3415 and 8.3420. So percentage increase, we need to find the percentage increase from 15 to 20 number of discs. So here it will be percentage increase, and that will be 8.3 minus 7.3 divided by the smallest value, that's 7.3 into 100%. And if you calculate it, you get a value of 13.7%. And with this, the percentage increase is calculated. Moving to part 3. Explain the relationship between the concentration of enzyme and mean volume of oxygen produced in 1 minute. So we need to now explain the entire relationship. Explaining means that whenever we are increasing the enzyme concentration, it obviously increases the volume of oxygen produced, because as the enzyme concentration has increased, it means that more active sites are now available, and now more enzymes can form complexes, that enzyme substrate complex, to produce more oxygen and eventually increase the volume of oxygen produced. So that's why increasing the enzyme concentration increases the volume of oxygen produced. I hope I have made my points clear. So write it down. Increasing enzyme concentration increases the volume of oxygen produced, because more active sites are now available, so more enzyme substrate complex can be formed. So more enzyme substrate complexes are formed to produce more oxygen. And with this, our question number C3 ends. I hope it was clear. Moving to part D. Suggest why the teacher measures the volume of oxygen after the first minute of the reaction rather than after 10 minutes. So when the teacher was calculating the amount of oxygen produced in the first minute, she was actually measuring the initial rate. And if she have the value for the initial rate, then she could use it to compare it with the overall rate of the oxygen produced. So there will be a fair comparison between the amount of oxygen produced or the rate of oxygen produced, so that she can compare the initial rate with the final rate, and eventually she can say that with time the rate of oxygen produced decreases, or with time the rate of oxygen produced increases. In order to say this sentence, she need to compare the final rate of the oxygen produced with the initial rate of the oxygen produced. So that's why she calculates the initial rate using the volume of oxygen produced in the first minute. So let's write it down. It gives a measurement for the initial rate because as time increases, H2O2 concentration decreases, so the teacher can make a valid comparison. So with this, our question number 6D ends, and also the entire paper for biology, paper 2, January 2021. I hope this entire paper was clear enough for you all to understand. If you have any questions that after watching this video you still didn't understood, you can write it in the comment section. I would really like to answer your comments. And if you want me to solve any other papers, you can also recommend the papers in the comment section or in our Facebook page Edexcel Easy, or you can email us to muhimaahmed01@gmail.com. And for this question paper and mark scheme, we need to email us again at the email address muhimaahmed01@gmail.com. All these screenshots will be added at the end of the question at the end of this video, and the screenshots will be added at the end. And if you need the paper, you need to email us, and all the links will be added in our Facebook page and group Edexcel Easy. So everything will be described below in the description box. So thank you for watching.